My name is Lindsay Huebner. I work in the corals program at the Fish and Wildlife Research Institute. I am a biologist here, and recently we have this project offshore from the Tampa Bay area to look at the coral and benthic communities in the eastern Gulf of Mexico. We have 15 sites that we're looking at, and these sites are a range of depth from around 30 feet or 10 meters to about 90 feet or 30 meters. And these sites are at ledge habitats. So these ledge habitats are kind of like this structural oasis in the middle of like an otherwise flat bottom. So these aren't reefs that are built by corals like you find in the Florida Keys. These instead are what I think used to be, old shorelines of the coast of Florida. So there's this like hard bottom, and then there's just this undercut ledge, and it drops, a little bit, maybe 6 inches at some sites, all the way down to several feet, and then there's soft or sandy bottom on the other side of it. And so these habitats create this vertical structure under which fish can hide, but we're also seeing that on the hard bottom side, there are a lot of stony corals, soft corals, different types of sponges, all sorts of things are really congregating on these habitats. So what we do is we go down to these ledge habitats and we lay out a transect line. So that's 50 meters we lay along the ledge, and that's going to allow us to see like what types of organisms are living like right on the edge of the ledge. And then we run several transects away from the ledge. So as you go from the ledge and further away from it, how does that community change? And is it a different community or is it really just the same at the ledge edge? And we use those to orient transects for both stony coral demographic surveys in which we measure all the species of stony corals that we find. For ones 4 centimeters or larger, we measure their width, their height, and also how they're doing. So are these stony corals bleached? Are they healthy? Do they have any diseases? Are they partly dead or are they totally dead? So we can get a lot of information about how these corals are doing just from these transects. And then on top of the stony coral surveys, we're taking photographic images of the bottom. And so we can see, in addition to the corals, what other types of organisms live at these habitats.
My name is Robert Ellis. I'm a research scientist with the Florida Fish and Wildlife Research Institute. For this project, I'm in charge of the fish survey component. We're really interested in the entire fish community as well, not just the groupers and the snappers that are swimming around, but all of the smaller gobies and blennies and the wrasses and all of those fish that might be more cryptic or might be hiding in little holes in the reef. The idea is that I'm going to count all the fish that are within 5 meters of me in a circle. So a cylinder that extends 5 meters away from me and goes all the way up to the surface of the water. This type of fish survey is used a lot, especially here in South Florida, to survey coral reef communities. So we've modified the design a little bit so that ideally the middle of the ledge goes through this sort of invisible cylinder that I'm in the middle of, and then I count all the fish that I can see inside that 5 meters. The species and number can tell us a lot about who's there, who's living on the reef, and how abundant they are. And we use that data to track both those short-term impacts from things like red tide and then the longer-term changes in the community that we might see by doing repeated surveys year after year.
Hi, my name is Christina. I'm a coral biologist at the Florida Fish and Wildlife Research Institute here in St. Pete. My main responsibility for this project is to kind of oversee the imagery archive. For the photographic transects, what we do is basically one person swims all of the transects that we lay out, taking images of the bottom. And then we take about 20 images per transect, so usually one every half meter or so. For the flyby images, we have to first set up the site. So that involves 2 different pieces. So we have the target, which is basically just a weighted down square that frames the rectangle of the transect. It has a color wheel on it, which helps us to color correct the image later on. And it has a little ruler on it. So it has a 10 centimeter marker, which helps us measure things on the mosaic later. We also have the elbow, which is basically just an L-shaped PVC. So we lay them out 2 on each side, and the goal with those is to get them on as much different height of the ledge as possible, so that helps us later to do rugosity measurements. The person who's going to be taking the images goes up about 2 or 3 meters off the bottom, and then they're facing straight down, and then they swim the transect and just continuously take pictures of the bottom. So we do 4 of those at each of the sites that we go to. And then what's cool is that we go back to the office, take these images, and you can enter them into a program that actually uses an algorithm to recognize the overlap in the images and stitches them together to make one really large mosaic.
The reason we chose this area in particular is because it's identified as a biogeographic transition zone. So we're in this sort of like switch area between subtropical and temperate. And this becomes important when we think about this habitat in the context of climate change. So if our waters are warming, then we might see a northward shift of subtropical animals which are typically found south of Tampa Bay, moving northward. And so when we look at all of those data together, we can see how these communities on the bottom at these ledges might vary by depth, and then also by north to south transition.
So one of the really cool things about this project is that it's linking together a lot of different data that we tend to collect separately. So we have everything from the benthic community, the corals and the sponges, and the anemones that are on the bottom. We're getting information about the physical structure of these ledges, and then we have the fish community data as well. And we're trying to link those things together into a full understanding of what these hard bottom ledge communities really look like. And understanding that will help us to predict and understand changes that might happen in the future.